mercury pressure, helps maintain the shape of the eyeball, much like inflated, much like a balloon, and then the light passes through that and arrives at the back of the eye, and with what you'll see through the ophthalmoscope, mostly just vascular area, right, gives it this reddish color, optic disc or the scalpoma or blind spot, blood vessels entering and leaving the optic nerve leading, and an area over here where there's not so many blood vessels visible, an area called the fovea centralis and the macula lutea, with macula lutea just a yellow spot, and we'll talk a little bit about the fovea in a second. If you look at the retina, the light in this case will be coming from the floor traveling to the ceiling, and you find that you have several layers of cells, the nerve fibers at the top, ganglion cells, like the final common pathway, they give rise to the optic nerve, an interplexiform layer, which is a layer of a lot of synapses, a bunch of cells called amacrine cells, then another kind of conduct information horizontally, another vertical conduction called the bipolar cell, an outer plexiform layer, another layer of synapses between the bipolar cells, some other horizontal cells, they too transmit information across the retina. And then, last but not least, at the back here are the rods and the cones, the receptors for the light, and pigment epithelium. So at first glance, it looks like the retina is built upside down. Because you've got the light coming in that picture that you've got your hand up that I just had, it's coming from the floor to the ceiling, which means that the light has to pass through the optic nerve fibers and all of the other cells. And as though nature has made the retina as transparent as possible, it's still, there is still some light absorbed. There's still some light refracted away from the pathway. So instead of passing directly through everything to the receptor cells, and even if you look, we'll get a little bit more, but the part of the receptors that absorb the light are actually back here, so it's actually got to pass through most of the receptor cells before it gets back here to where the light is absorbed. So in some ways it looks like the retina is built upside down. However, it really is the best compromise that you can get to accomplish the job that you need to do. The receptors, especially the outer segment. Everything, by the way, is measured from the center of the eyeball as if it was a sphere. So that would be, my finger here would be the center. So if you're going to the back where the retina is and you keep going, it's out. If it's this way, it's in. So anyway, the outer segment, we'll talk a little more about that a little later. Anyway, so you've got this business of the receptor part, the active portion of the receptors embedded in this pigment epithelium. It's just a single layer of cells that contain a pigment called melanin. It's the same brownish black pigment that you find in your hair, the iris of your eye, the epithelium or the dermis of your skin. So they're around those photoreceptors, the active outer segment of photoreceptors. And it does two things. One is that it absorbs the light not absorbed by the photoreceptors. You don't particularly want the light coming in, passing through all those cells, some of it being absorbed by the receptor cells hitting the back of the eye and bouncing back. Hits the back, bounces back up the mirror. It would blur the image. Make it more confusing. So you put the pigment at the back, so any of the light that isn't absorbed by the rods or cones, depending where you are in the retina, it gets absorbed. So it comes in, gets absorbed, either by the rod or the cone, or the pigment epithelium, no reflection back. Now, there are some animals, like cats and stuff, that have a specialized portion in the retina to act as a mirror to enhance their night vision. But we're sticking to talking about people here. Okay, so yeah, in case somebody thinks about their pet or something, yeah. I am aware of that, but we're going to stick with people for the moment. Okay, so that's the first thing. So if you had the rods and cones up in front, then you have to figure a way of putting the pigment behind, but not block off all the processing cells, and then, so it's one compromise. Okay, the other is that the retinal pigment epithelium also seems to have a very key role in transporting nutrients and ions to the photoreceptors, and is absolutely required for the retina, and we haven't talked about these chemicals, but we will, is absolutely required for the regeneration of 11 cis retin tau, and the absorption of light, not so much for I said that. Okay, so it's doing two things. One is the pigment epithelium in the back there absorbs extra light, and second, it has a metabolic function in that it helps regenerate some of the visual pigments. Okay, so we've got to get some of the anatomy out of the way before we can talk about all the physiology. This is a schematic diagram of the area of the retina that contains the fovea. And the fovea is the area of highest visual acuity. You have all your cones are packed into that area. And it's the area where when you look to focus your concentration on something, Brad Pitt, Angelina Jolie, or whatever, that light from that you're focusing your attention on is automatically, if everything's normal, focused on the fovea. Well, the fovea actually looks like a little bit of a valley, because if you remember this stylized diagram of the retina, you've got all these cells here. These are all processing cells. They're receiving information flowing back from the receptors back. And so what nature has done is say, well, we're going to try as much as we can to make the link between the light coming in and the receptors where the action takes place as direct as possible. So essentially what's happened is all those processing cells have been shoved aside. So they're, as the diagram here, stylized diagram tries to show you, there's not much between the light coming in and the rods and the cones. So yeah, again, nature made this compromise. It couldn't do it for the whole retina, but for one area where you're going to concentrate your vision, we'll shove all the plumbing and processing stuff to one side. So there's a direct shot of light directly to the receptors. But you can only do it in a restricted place. So the fovea is this specialized area. Okay. We do a few facts and figures about the receptors as we start to build up how the system works. You have the rods and the cones, and this, I'm sure, comes as no surprise to anybody. They're named from their anatomical shape. The rods look like rods, and the cones look like inverted ice cream cones. All right, so there's about 100 to 125 billion per retina. It's a guess. I don't think they found a grad student to sit down and count them all, but it's a guess. Based on how well packed they are in a small area, and then you can extrapolate. They all contain the same visual pigment called rhodopsin, which again I'm sure you've heard of. So you see only shades of gray when you're using the rods. They have a very high sensitivity. They say that if you get 2 photons striking the same cone within, I forget what tiny bit of time, you'll see it as a spotlight, so they're very sensitive. So they have low acuity pathways. What that means is you got lots of rods feeding on to a few, ultimately a few ganglion cells. In other words, they take a lot of information over the area of the retina, summate it, and feed it through. So you get very high sensitivity. You can see more or less, depending on how dark it is, you can see it, look around. But it's not sharp and distinct, so it's low acuity. It's obviously what we use at night. Because these two kind of go together, low acuity pathway, much convergence in the retinal pathways, lots of rods signaling onto fewer and fewer processing cells. And they're most numerous in the retinal periphery, that is, they're not in the fovea. As you go from the fovea outward, the number of cones goes down and the number of rods goes up. The cones, it's about 6 million per retina. They're responsible for color vision because each cone contains a choice of one, again, normal vision, one of 3 pigments. It's either a pigment that absorbs maximally in the blue, a pigment that absorbs maximally in the green, and a pigment that absorbs maximally in the yellow area. But that one happens to be very broad and also covers orange and red. So blue, green, and yellow. But most books say because it's mostly used for the longer wavelengths, most books will say blue, green, and red. But if you look at the actual absorption spectra, which you probably have no overwhelming desire to do, you see that it's maximally toward the yellow. Okay, so each cone has one of the 3, not all of the 3, not 2, 1, but it's going to be blue, green, or red. Okay, they have low sensitivity, that is, it takes a little more energy to activate them. They're high acuity pathways, that is, there's not a lot of convergence, that is, you almost have a direct line of cone, bipolar cell, ganglion cells, so you have the minimum number of processing cells in the pathway. No surprise here that it's what we use in day vision. Little convergence, and they're concentrated in the fovea. Okay, now, this is where we're going to talk about what we call the primary transduction process. That is, how do we take photons of energy and convert it into an electrochemical signal that the brain can handle. Okay, so you're sitting in the dark, bottom of a cave, somebody watching your closet, whatever. Okay, you're sitting in the dark, no light. There's in the rods, which are ones primarily that we, because they're so easy to find and so many of them are ones that most of the studies have been done, but it pretty much holds true for the cones as well. There's you find a high concentration of cyclic GMP, one of the second messengers that are used. A lot of sodium channels are open in the outer segment, which means that the membrane is depolarized. Okay. Which means that calcium channels are open in the synaptic terminal, which means that there is an increased release of the inhibitory transmitter. That transmitter has to be glutamate. Which means that if you've got an inhibitory transmitter, since the usual pathway of receptor cell bipolar means that the bipolar cell is inhibited. Remember, we've got information flow vertically and horizontal. Receptor, bipolar, ganglion cell is the vertical pathway, taking the information from the back of the retina where the rod was activated out to send it to the brain. Horizontal means going across the retina is the horizontal cells. That's how they got their name. Sometimes they're not too inspired to name things. Sometimes it actually works well because it actually tells you what they're doing. And the amacrine cells also kind of transmit information across the retina as well. So you're inhibiting the vertical pathway here, bipolar cells, which means there's no action potential in the ganglion cell. I'll just face the sentence then I'll get you. The ganglion cell is the only cell that generates action potential. All these others are working on slight changes in membrane potential. Yes?
Did you get the inhibitory compound that was used for glutamate?
Glutamate.
I thought glutamate was excitatory.
Is which?
Excitatory?
You know, any transmitter could be either one. It depends on what the effect is when it binds to the receptor on the receiving end. What does it activate? If it opens sodium channels, it could be excitatory. If it closes them, it would. So yeah, it's very site specific. You know, you can look around and you find what one transmitter is active here could be over the other. One you'll see. Have you done just an example? Have you done digestion yet? Okay. It's not a neurotransmitter, but the fact's the same. There's a hormone called cholecystokinin. Cholecystokinin is released in the gut when you have fat or protein in the context of the gut. When it stimulates the presence of fats and proteins in the diet, stimulates release of this cholecystokinin, which is a hormone. It's also a neurotransmitter in the brain as well. When it goes to the gallbladder, it tells the smooth muscle in the gallbladder to contract, but it also tells the sphincter vodi, which is a donut-shaped ring of smooth muscle that opens and closes the bile duct. It tells the bile duct to open. Same hormone. Gallbladder, smooth muscle, contract. Regular smooth muscle at the sphincter vodi to open. So that's probably the easiest example of it, because everybody kind of knows what having gallbladder. You go into some more obscure places with that. And so it's the same thing. In this particular case, it inhibits the bipolar cell by its actions on the postsynaptic membrane. Okay, so there's no action potential ganglion cell, no action potential lateral geniculate nucleus. Lateral geniculate nucleus is in the thalamus, and the thalamus is called the gateway to the cortex. It's in the brain there. Any sensory experience that you're going to become consciously aware of has to make a synapse in the lateral geniculate body. So all that's just emphasizing no message is going through. Okay? Because some people like to learn with words and other people learn better with pictures, we have about the same thing there. You got the light will be coming up here, you got the cycle of GMP, these sodium channels are open, so you get this current flux of the cells are polarized. I mean, you call it sodium potassium flux, which should always have to be now. So then this is releasing glutamate down here and inhibiting the bipolar. So this just says sodium is diffusing in, cyclic GMP or channels are open, sodium channels closed, you lose the cyclic GMP. So same thing I said in one way on a flow chart, give you a picture as well. Okay, so that's what's happening in the dark. What happens when you then have some light come in? Okay, so light comes in, and get to use, I love it, get to use all these lovely chemical terms now. Isomerization of retinal, which is derived from vitamin A, so eat your carrots, he forgot what your mother told you, and absorbs the light. Okay? We'll give you a little bit more information about retinal and retinol in a moment. So you change, essentially what you're saying is you change the shape of retinal from one form to another when you absorb light. So you're putting energy in and you change the shape of the molecule. It's a real relatively simple basic chemistry. You got one chemical structure, you put energy in, the molecule absorbs it, and it might change shape. This change in isomerization of retinol stimulates the activation of a G protein called transducin. Transducin. The transducin activates a phosphodiesterase. This activated phosphodiesterase breaks down cyclic GMP, which is what normally happens to a second messenger anyway. That decreases cyclic GMP, closes sodium channels. This leads to hyperpolarization of the rod, which closes the sodium channels with the synaptic terminal. Kind of what happens in a lot of nerve cells. This decreases the release of inhibitory transmitter. Bipolar cells are disinhibited, that is, they were inhibited, and now you're inhibiting the inhibition, which is essentially the same thing as activation. Okay, so it's in effect, they're excited. So the bipolar cells get a gradient potential, no action potential. These guys only signal by slight changes in membrane potential. Remember that whole business I gave you about EPSP and IPSP? But that's enough to send a signal to the ganglion cells and depolarize the ganglion cells. And so you generate an action potential, and the action potential is then traveled back. Now, why do the cells only use minor changes in potential to signal? Because you have 125 million rods, 6 million cones. I didn't tell you how many bipolars, amacrines, and well, ganglion cells is about 1 million. So you're looking at a huge number of cells packed into a very small area. If everybody was generating action potentials, there would be a possibility of the current flows on these small diameter fibers, the action potentials coursing down one to depolarize another one, and you'd get all kinds of action potentials generated that the body couldn't interpret. So if all you do is have everybody release their transmitters and stuff due to minor changes in membrane potentials like EPSPs or inhibited like an IPSP, there's less opportunity for crosstalk, less opportunity for one cell becoming active and inadvertently activating another one, because you have so many nerve cells. These are all essentially nerve cells packed in a very small area. So you can get over it or get around the problem by just having changes like an EPSP to generate a potential, IPSP to regulate whether or not a transmitter will be released or not. Okay? Okay. So this is, before anybody panics, no, you do not have to memorize the chemical structure. This is the kind of thing. It starts out like this. You put the photon in, it looks like that. It, remember the old light switch? You flick. It's almost like that. It's kind of like this bent portion of the molecule, and you put the light in, and it's like you flip the switch up, and this part shifts from kind of this downward curve into straight across. So that's the isomerization that we're talking about. And so this rhodopsin is made of retinal and the protein opsin. And retinal is the aldehyde formed by it. It comes in an aldehyde, it comes as an alcohol, and it comes as in an acidic form as well. Most of them the liver can interconvert for you. So when you're eating your carrots and other colored vegetables, you're taking in a form of vitamin A, depending on what the vegetable is, either retinol, or retinoic acid. Just to show off that I did my homework and remember all three, the needs all three. Okay? So, the all-trans retinol is reduced to all-trans retinol. Hopefully that's what it says on the picture as well, when it absorbs the light. And this then leaves the cell, leaves the rod, and is transported in case you go home with all this nice, give you some nice polysyllabic terms to impress your friends and neighbors that you're getting your money's worth here. So this all-trans-retinol is transported itself by interphotoreceptor retinoid binding protein. Very impressive to your young siblings. IRBP and extracellular carrier protein is just like the plasma proteins in the blood that bind up stuff and transport it around. To the overlaying retinal pigment epithelial cells where it is reconverted to retinol, alcohol, then to retinol and returned to the rod where it spontaneously rebinds with the opsin, and voila, you are back in business with active rhodopsin for your night vision. The cones work pretty much the same way. The only difference is that the pigment portion is slightly different than rhodopsin. So the blue pigment's a little different than the green pigment's a little different than the yellow pigment. But that's pretty much about the only difference. Now, this is all swell. Some of this thing. You know, we've got these rods and the cones. We told you that what they do in the dark, they're giving the pathway and the light. You get you inhibit the receptor cells and turn everybody else on. And these changes in membrane potential go from the receptor to bipolar to the ganglion cells, and the action potential go back. Now, we only have 3 lectures, so I can't go into all the wonderfulness that happens as these action potentials go back, so we won't talk too much about the thalamus and the cortex. But what we will do is we will try and put together for you what these little signals in these individual rods and cones are doing. And this means you have to have the concept of what's known as a receptive field. And a receptive field is an area on the retina which, when stimulated, causes a change in the output of either a particular bipolar cell or ganglion cell. Some people like to record from bipolar, some people like to record from ganglion cell. So it is a distinct area. You can map it. Wherein you put a spot of light and whichever cell you like, I don't think in a ganglion cell, but that's just me, is either activated or inhibited. So it's an actual area on the retina, you can map it. That when you stimulate light in that particular receptive field, a particular cell is going to respond. Because it's easier to look at one at a time. Anyway, in humans, the receptive fields tend to, they're not all exactly concentric circles, but by and large, they're similar. Some of them are nice and round, some of them may be slightly similar. But anyway, and so what you have is these areas. So what the researcher does goes in, sticks his microelectrode into a ganglion cell, let's say. And then the animal that he's using is fixed and staring at a screen, and he basically has you either use a white spot or a black dot. You're using the white spot, use a black background, vice versa. And you basically have it track across the screen. And when all of a sudden it hits the area out there in the real world, that particular ganglion cell is receiving information from a group of receptors that will respond. Remember, it's only the receptors that respond to light. You can shine however bright a light you want on a bipolar, a gamma current, a horizontal, or a ganglion cell. It doesn't do nothing. Okay? So what we're doing is we're putting a spot of light in, and it's activating receptors, and we're watching what the response is. It's sort of like a black box. You put something in, you measure what comes out. You may not be totally sure what's happening in between. Okay? So what they find is that Human receptive fields tend to fall into two categories, one called on-center, off-surround, the other called off-center, on-surround. In other words, reciprocally different from each other. Very simply, it says that if you put a spot, and it can be a spot of light if you're looking at a dark background, it can be a black spot if you're using a white background. Anywhere in the center area, you'll get nothing happening. You turn the light on, you get a burst of action potentials, kind of fades out a little bit, and then you turn it off, and nothing happens. If you put the spot out in the periphery, nothing, this is just a random action potentials that occur. Boom, turn it on, nothing happens. You get inhibition, you turn the spot out here, and you get a brief burst of action potentials, and then nothing. And so it's just the reverse here. Nothing when the light goes on in the off center, a little bit when you turn it off, put the light in the surround, and you get a lot of action potentials that are disappear when you turn it off. Okay? Now a little more detail on this type of thing is something like variation on that. It says, okay, you got an on-center ganglion cell. You put, you got an area that's excitatory and an area that's inhibitory. So you put a spot of light in, you get action. Put a spot in the periphery, you get inhibition. The more of the center that's illumined, the more action potential per second you get. The same thing, the more of the surround that's illuminated, the more profound the inhibition, and the more action potential when you turn it off. Diffuse illumination, that is illumination over the whole of the receptive field, gives you a modest increase in action potential. It just kind of tells you that whatever the surround is tends to slightly predominate. And you go down the same thing with the off-center, on-surround. The light in the center, inhibition, turn it off, it's an excitation. Light in the surround, action potentials. The more the surround illuminated, the more profound the inhibition, the more action potential when you turn it off. The more the surround illuminated, the more action potential, diffuse illumination. Not too much, but when you turn it off, get a bit of a burst. So this is telling you that by and large, and this is a simplified version, that by and large, you're not interested in absolute intensity. This doesn't tell you particularly how bright the world is. Basically telling you where a stimulus is. Now, they have been teasing out more and more of the retina. And they have found that there are some ganglion cells that seem to be wired up to their receptors in such a way that they do act as luminous detectors. In other words, they do act like a light meter and do give you some kind of information on how bright it is. But most of them seem to send their information to subcortical areas where reflex activity is taken. So you're not really consciously aware of the output of these cells to try to get that information in other ways. So it's mostly pupillary reflexes and some other unconscious reflexes. Now, this on-off business is kind of the foundation. They have now found that there are some receptive fields that are color-coded. That is, they may have a red center, blue off surround, various combinations between the blue, the green, and the red. And so that some are telling you that there's a certain color in the receptive field in the center and some other color hitting in the surround. So we're finding that you got all this simultaneous processing going on. I talked about on Wednesday. You got some here telling you luminous, how bright it is. Some are telling you where something is by these spots. Some use a similar sort of receptive field combination, but are connected up to different colored cones so that you get color information. but this is kind of the basis for most, except that one. Okay, so this is a demonstration of why I didn't go into art. I think you can tell this is hand-drawn, and it is. But to try and give you some kind of a feel, I mean, this is it, folks. Those two types of receptive fields are what you've got. If you're a frog, there's about 5 different receptive fields, but then the frog doesn't have a whole lot of brain space, so it's got to wire things up differently. Ground squirrels have receptive fields that preference to tell them which way the spot's moving. If it's going this way or this way. We've got lots of brain capacity back here in the visual cortex, and our receptive fields seem to be correspondingly simple because we've got more space to process information. Okay, so all I've done here is say you've got 2 receptive fields here. They're not lined up, and they're deliberately overlapped. They are not lined up like little nice little circles across the retina. They massively overlap. Okay, they're not just a bunch of circles like this. They, look more like that. Okay? So, not being sharp enough to do 5 or 6 all at once, 2 seems to get the point across. So what we've done is we've got, say here we got 2 off-center, on-surround receptor fields. And I say if you have a spot of light here, how is each cell going to respond? Well, this cell here, everybody with me on this one? The spot's here, so it's in the center of that receptive field, so you're going to get it in vision. But at the same time, the spot here is in the surround of this cell. I guess I couldn't afford a 3rd color. You might tell, okay? So that the spot, that same spot is now in the surround of this, so you get an on response, right? And then I guess I ran out of acetates as well when I was doing this because I still want some overhead. But 2 on 1. So this is the same thing except now they're both on center. And it's the same concept. One cell is going to be activated because it's in the center. The other one's going to be inhibited because in this case it's in the surround. And then I did an on-off one here where this cell's on, so it's going to be activated. And because this is an off-center, on-surround, that spot's going to do the same thing and activate that cell. Now that's the information you get. That is the information that your retina is providing you. Now, we try to put together a wiring network. How do you explain how you get those on and off type responses? And that's what this is. It basically says, oh, you got all these rods here all packed together. And all it says is that there's a group of rods that are all wired together to feed on the bipolar cell. All and you put the light in the center, and you hyperpolarize the rod, which disinhibits the bipolar cell so it becomes active. It comes right out of, I think this is one year's recommended text. And in this case, if the spot falls on these other side, well then these rods become active, but now they have base synapse on the horizontal cells, which hyperpolarizes the bipolar and gives you the off response. Okay, so these are all the same. They're just colored just for your make things a little clearer. So what they're saying is if the light is falling on a bunch of rods that form the center of a particular ganglion cell or bipolar cell receptive field, they're wired up in such a way that the bipolar is directly wired to the receptors, and you get activation. Others, because you're again you can invoke the horizontal cell because you're bringing information from across the retina. So they get interspersed in that outer plexiform layer rather. And so when the light falls in the surround, the bipolar, the rods do their thing, but now they're feeding onto some horizontals that turn off the bipolar cell. And if you look at the terminal endings in here in the retina, you see that there's and at the base of every rod, there's all kinds of synaptic endings from horizontal cells and bipolar cells. So it's not a stretch to say that one rod can feed numerous bipolar cells, either direct or through those interneurons, the horizontal cells. Okay, move along. So let's say we got some light that hit into the center of an on-type receptive field. I guess I should back up and say this system works really, really well for where edges are. Now, those bars are just an absolutely glorious stimulus because those edges, because there's a nice contrast between this off-white paint and this dark edge. And that edge, the contrast between the paint and that edge is a really good stimulus to those receptive fields. So it really is a nice system that tells you where changes are. All of you wearing stripes out there, be they vertical or horizontal, excellent stimuli for these receptive fields. Okay, so we think that these receptive fields help the body or the brain put together a picture of where there's contrast and changes edges in the receptive field. So even all these little lines on the tiles where they're dark and light, very good stimulus. Now, we got all these action potentials and all these ganglion cells are all cranked up, generating action potentials like crazy. And the axon from the ganglion cell is the optic nerve. So there's real action potentials because you've got to transmit it from out here way back to the middle of the brain. And everybody may hope has had the opportunity to take a human brain and you turn it over and you see this great big white X on the bottom. That's the optic chiasma. That's where the two optic nerves come together and then the optic tract leaves. Everybody says, oh yeah, they cross over. Well, I wish it was so simple. It isn't quite that way. My attempts at art, that is supposed to be, that little inverted V is your nose. Okay, well, it's good to have a reference point. So what happens is, as I say, if you look out there in the world, if you take a look at each eyeball, you can draw a plane through and divide it in half, a vertical plane. One side is going to be closest to the temporal bone, that's the thing on the side of your head. The other half is going to be close to your nose. So we call it the temporal retina and the nasal hemiretina. Okay? So if you look, what we're saying is that these that are over on the temporal retina here, in the left eye, come down to the optic chiasma, but they don't cross over. They stay on the ipsilateral side. Ipsilateral means on the same side. Okay? The ones on that would be in the temporal hemiretina, come down to the optic chiasma, but they do cross over. This here is the lateral nasal. Okay? And then the other thing, the nasal retina here cross over, and the temporal retina on the other side. What the body is doing is bringing those ganglion cells, subserving those receptors on each eye that see the same thing out there in visual space, bringing them together. So it's a pretty nifty trick when you think about it, because this develops in utero. There's a million fibers here and a million fibers here, but they already know that they're either on the temporal or the nasal hemiretina, and know that when they travel back in utero and you're developing, they know if they should cross over or not. It's a pretty neat trick when you think about it. 2 million fibers knowing where they're supposed to be. Anyway, so all that does is it brings those particular cells that are seeing the same thing out there in visual space together. There's further consolidation in this lateral geniculate body, and then it goes, all the information goes back to the occipital cortex, or the visual cortex. Okay. Where we have areas of the brain that seem to, again, we don't have time with only 3 lectures to go into it, but what happens is that we know that there are areas in the cortex that act as simultaneous processes. There's some areas that the cells in the visual cortex might respond when whatever it is out there in the real world is moving. There are others that respond to the shapes or forms, that is, sharp edges. There are some cells in the visual areas of the visual cortex where the cells receive input from both eyes and give you your perception of depth, because each eye sees things slightly differently. And lastly, there's a large area of the cortex where the cells respond to color. So you got, essentially, we think about 4 simultaneous processes going on. Motion, shape or form, depth, and color. Okay? So those are the 4 main aspects of what you're looking at there. And somewhere in that visual cortex, all 4 of those aspects are put together and give you the complex color, the kind of visual world that you see. We don't know exactly how it all comes together, but we can tease out separate bits of it and make some guesses. But that's pretty much it. Well, I got 2 minutes. Somehow I don't think I can get through too much. Next time, Monday, we will start hearing, and we will take a look at how a different sensory system using an entirely different form of energy encodes in